Right, good morning again. So I have the pleasure today to have Dr. Robinson visiting the lab here, and he will be presenting an interesting topic in the area of hydration. And I love the title, From Neighborhood to Nephrons, so trying to cover everything we know, anything in between. So a couple housekeeping things that I wanted to remind you. If you want containing education points for the three associations that we are affiliated with, we will post the links in the chat later on during the presentation, so make sure you click the link to claim the CEUs. And also would like to thank Unilever Wellbeing Commons for sponsoring all the water talks for the next. We will have small break for July, but we'll have the coming back in August with the next one. And we will continue for the rest of the year with more interesting topics. So Dr. Robinson, thank you again for coming to Phoenix, and looking forward to your talk.
So thanks for that kind introduction, Stavros. Thank you to everyone here in the room and everyone who's tuning in online. So the title of this talk is From Neighborhood to Nephrons, Hydration Practices, Social Context, and Biomarkers in a Racially Diverse Young Adult Cohort. So first off, disclosures, I am an associate editor for the Journal of Applied Physiology. I won't be presenting on any work that we've published in the journal today, and I don't have any financial disclosures. In terms of objectives for the talk, we'll describe racial differences in cardiometabolic health. We'll examine racial differences in hydration status and fluid intake. We'll identify social determinants of health that may contribute to hydration disparities. We'll discuss the relation between hydration and kidney health briefly, and discuss implications of racial hydration disparities. All right, so getting into my background, I'm interested in hypertension and cardiovascular disease, and my interest in this field led me into some of the work that I'll present today. So I'll start a bit with my background and how I got here today. So hypertension is the leading modifiable risk factor for cardiovascular disease, which is the leading cause of death in the United States. We know that it affects 1.3 billion people globally, and it accounts for 10 million deaths globally. So on this data, we're looking at age on the x-axis and percent of the population with hypertension on the y-axis. You can see there's an age-related increase such that in our lifetime, over 9 in 10 Americans will develop hypertension. So while hypertension is a major public health problem, we know it doesn't affect everyone equally. So this is an older ethnographic study comparing hypertension prevalence across several different countries. One thing I do want to note is that the guidelines for hypertension changed in 2017 to be more stringent. So the overall prevalence is actually higher than what's depicted here. But what I want to draw your attention to is that Black Americans have some of the highest hypertension rates in the world. And one thing that I'll point out here is that other countries with a high degree of West African ancestry have much lower hypertension prevalence. So you can see Jamaica here and Nigeria, which has a quite low hypertension prevalence. Now, if we look at the US specifically, this is data from NHANES. Irrespective of biological sex, we can see that Black adults have the highest hypertension prevalence in the United States compared to other racial and ethnic groups. And so that gets me into my initial interest and how I eventually started getting into hydration disparities. So during my postdoc, I worked with Dr. Bill Farquhar, who you can see here at the University of Delaware, and I helped on a study where we did a hypertonic saline infusion. So for a bit of background, you can place an intravenous catheter and you can infuse hypertonic saline. So in this case, 3% sodium chloride. This is a model that you can use to kind of roughly mimic what happens with sodium loading. And so in this particular study, we took samples at baseline, 20 minutes, 40 minutes, and 60 minutes. And so this is the data where we compared Caucasian individuals and African-American individuals. Serum sodium at the beginning of the study was actually quite similar. However, over the course of the study, there was a larger increase in serum sodium in Black adults. And for any given increase in serum sodium, we noted that there was a larger increase in systolic blood pressure in the Black adults. So that led me to develop an acute soup study, which is I was able to secure funding for a career development award. And I was really interested in some of the factors that could lead to racial disparities in response to the sodium load. So in this particular study, as I mentioned, we used soup. So there was a low salt soup condition, randomized crossover design, and a high salt soup condition. And so as I prepared for this study, we did a separate acute soup study, irrespective of race. And this is just to kind of show the audience here that similar to the hypertonic saline infusion, when you give people a high salt soup meal, there is an increase in serum sodium. There is some nuance to this, like depending if you drink water along with the high salt meal, but in general, you can increase serum sodium, not quite to the same extent, but it's a good model to look at the acute effects of salt loading. And so we are particularly interested in racial disparities here. Now, while doing my lit review for the K Award, I came across this concept of the slavery hypertension hypothesis. This is actually something I first heard of in high school from a history teacher, but it became popularized in the 90s, mid 2000s. There was an episode of Oprah where Dr. Oz was on, and he kind of peddled this myth, if you'll call it that. But there's a couple of studies that have debunked this hypothesis. And basically the idea was that early investigators back in the 70s and 80s thought that Black Americans faced a number of selection pressures that led to greater sodium retention, and that contributed to the differences in salt-sensitive hypertension. So to get into that a little bit more, the three key selection pressures were thought to be the hot and arid African environment, and lack of salt. However, historians have pointed out that there was a robust salt trade in West Africa, the transatlantic slave trade, so no one would argue that that wasn't a terrible event. However, the slaves were viewed as a commodity, and they were given salt, and the slave traders were trying to keep them alive, even though it was, terrible conditions. And then with New World slavery, less so were effects of water and electrolyte imbalances. It was more major causes of morbidity and mortality would have been taking people from Africa, displacing them into America before modern public health, and they didn't have any of the immune adaptation for the new environment. And there's even data to suggest that salt-sensitive hypertension is not really reliant on sodium retention per se. So this is data from Dr. Curtis Morris's group. They were at the University of California at San Francisco and did a lot of work on salt-sensitive hypertension in Black adults in the 90s and 2000s. And so here, this is within an African-American cohort, and we can see the main difference between salt-resistant and salt-sensitive participants was changes in renal blood flow and an increase in renal vascular resistance, which would reflect that an increase in systemic vascular resistance really drove the difference in blood pressure responses to sodium. And so we do know that West African ancestry is highly prevalent in the United States in Black adults from the transatlantic slave trade. However, data suggests that West African ancestry is not a major predictor of blood pressure or cardiovascular events. So as just one example, a few years ago, there was a secondary analysis from the SPRINT trial, and we can see African ancestry here on the x-axis, if anything, was inversely related to the hazard ratio for composite outcomes. So in this case, that would be things like myocardial infarction, stroke, or cardiac arrest. So that led me to be more interested in social determinants of health that could be contributing to some of these health disparities, not only with salt-sensitive blood pressure, but health disparities more generally in cardiometabolic health. So for anyone who's interested in this kind of passively, I would recommend this book, The Color of Law by Dr. Richard Rothstein. He's a lawyer at Yale. I also have a QR code here that leads to one of his YouTube talks that kind of concisely goes over what's covered in the book. But what mainly is covered in the book is racialized housing covenants and redlining. So redlining was a practice from the Homeowners Loan Corporation following the New Deal in the 1930s. And so they would grade neighborhoods from green to red or A to D. And the red neighborhoods or the red-lined neighborhoods were neighborhoods that had a higher proportion of Black individuals or immigrants. And so those were basically zoned off as like, you don't want to invest here. We don't want to give mortgages here. We don't want to build up the infrastructure and put good schools here and things of that nature. So for decades, there was disinvestment in Black neighborhoods or minority neighborhoods. It legally ended in 1968 with the Fair Housing Act, but the legacy of that still persists today. So the graphs that I'll present here on this slide are from a story in the New York Times a few years ago. But for example, here we're looking at median household income, and The blue dots represent majority white communities. Purple is majority Black communities across numerous cities in the United States. And so we can see a couple of prime examples here would be Minneapolis and San Francisco, but there's huge disparities in terms of median household income by race. There's also large differences in median home ownership. So again, for several decades, it was harder for Black families to secure mortgages or to secure insurance for their mortgages. So beyond that, we also know that major health organizations such as the American Heart Association also recognize the important role of social determinants of health. Just to kind of clarify what exactly that is, healthcare and access and quality, built and social environment in the neighborhood, economic stability, and educational access and quality are all major factors here. But there's been a number of reviews and position statements that have covered the importance of social determinants of health and cardiovascular disease and disparities. All right, so in building on my background with social determinants of health, when I took my first faculty position at Auburn University, I reached out to Dr. Thomas Fuller Rowell. He is a social psychologist interested in population health, and he had an R15 grant that was focused on neighborhood context and social support and how that influenced cardiovascular health in young adults, so college students. And his lab did a great job of things like assessing neighborhood throughout the individual's lifespan, adverse childhood experiences, things of that nature. And our lab focuses more on applied physiology. So the idea here was to bring participants who were in his study back to our lab and do some deeper phenotyping. So our particular interests were in parental education, family income, neighborhood environment, which I'll touch on more in the next slide. And then in our lab, we focused more on health behaviors as another exposure. So diet was assessed with 3-day food and fluid diaries. We measured objective physical activity with ectographs. And we use actigraphy to measure habitual sleep patterns. So once in the lab, we assessed vascular function, circulating biomarkers, and 24-hour urine samples, which is most of the data that I'll present today. Okay, so for neighborhood deprivation, this was the major social determinant that we were interested in. So we used the Area Deprivation Index. This is an index that was developed by the Center for Health Disparities Research at the University of Wisconsin. And so it's a composite score of 17 different indicators that characterize neighborhoods. So it includes median income, education attainment, employment status, and housing quality is just a few examples. So here I'm showing the state of Indiana where I live now. And you can see these colorized zones. So a darker red indicates more disadvantage, blues indicate less disadvantage. And you can break the data down by state deciles, which you can see here, or national deciles. And that's what we used in our study because we were studying college students. So they were from all over the US. This is Indianapolis, Indiana, so our capital city. Fairly similar to a lot of big cities around the country. Throughout the 20th century, there was kind of some disinvestment, some flight away from the inner city centers. So you can see within the city, there's a lot of deep red here, and then the surrounding suburbs, you can see a lot of blue. Anyways, in spite of those colors, we can also operationalize this data with a score. So we score deprivation from 0 to 100, and the higher the score, the more deprivation. So early on in the study, we made an observation. This is Sue Lim, a former grad student, holding one of our urine collection containers. And early on, we noticed that a lot of our Black participants were bringing in the containers with about 1,000 mLs or less of urine. This was an early observation made by my master's student, Alex Barnett. He'd done a lot of work with urine flow rate and urine volume from secondary analysis with our samples in Delaware. So he thought, is something wrong here? Are the participants maybe not collecting urine the way they were instructed? So we weren't sure if it was potentially that or maybe just noise from the first few participants coming in. So Alex made that initial observation and it became part of his master's degree project. And then Mason, who's here with us today, was also at Auburn. He joined Auburn for his PhD while this study was underway, and he helped present and pull together some of this data as well. So I'll give him a quick shout out. Here's Mason presenting a poster about 4 years ago at Kidney Camp. He actually won an award for that poster. But to get back on track, What we ended up doing was a lit review to determine if there were well-documented disparities in hydration status. And so this is data from NHANES, over 8,000 adults aged 20 to 40. And in this particular analysis, they use the urine osmolality value of greater than or equal to 800 to define inadequate hydration. And it's a huge sample. This is rich data, but there is a limitation that it's a spot urine sample. So inadequate hydration, we can see 27% of white adults fell into that category. And then pertaining to the health disparities, we see it's nearly 40% for Black adults and Latino adults and 27% for other. So our next question is, why does this disparity exist? And there's a couple of really well-documented examples of why predominantly Black communities may distrust their municipal water system. There's a really interesting book about this, published a handful of years ago now, called The Poisoned City by Anna Clark. So if anyone's interested, I'll put another QR code here. She gave an interesting seminar at Harvard several years ago now that kind of summarizes what's discussed in the book. But briefly, Flint was a booming town in the early 20th century. So there was a lot of manufacturing from GM. And then as the century progressed into like the mid 20th century, GM ended up leaving, a lot of the jobs left, and the population of Flint shrunk considerably. So it's like a classic Rust Belt town when manufacturing left. And you can see some houses here that have clearly been neglected. And that was kind of the story of Flint well into the late 90s and the 2010s. And so here, I want to depict this picture here of Flint, and you can see the Flint River. So in 2014, the city was under emergency management from the state, so they didn't have like a permanent mayor who was making the decisions. And so the emergency managers made the decision that Flint was going to switch from the Detroit water system to their own water system, but temporarily they were going to use the Flint River. So they powered up their water treatment plant, switched to the Flint River, and for reasons that still aren't really clear, they didn't add any anti-corrosives to the water. And so lead began to leach off from the old kind of outdated water pipes, and it was making kids sick. It was making a lot of people sick. So obviously it led to a state of emergency, and that was very well publicized. More recently, Jackson, Mississippi, they've had poor water infrastructure for a long time as well. But in 2022, there was a crisis where over 150,000 adults didn't have safe water to drink. So there was a number of boil water advisories, and people were relying on bottled water for things like even washing and brushing their teeth. So these are just two really well publicized cases of that. But we know around the country, if people think their water tastes bad or if it smells funny, they're not going to want to drink that, and they're not going to want to use it to make things like tea or coffee or different prepared beverages. And we know that almost half of US adults, based on this survey of over 4,000 people, are either neutral or disagree that their water is safe. So this is a pretty widespread problem. And in terms of implications, we can see that the odds of drinking less than 1 cup of tap water per day are threefold higher for individuals who disagree that their public water is safe. Just in full transparency, these data are a little less compelling for total water intake in sugar-sweetened beverages. So I don't need to tell this group here that hydration is important for health, but just in case, we will make the point here. Underhydration here was defined as high serum sodium or a high urine osmolality. In this particular analysis, over 60% of adults were defined as having underhydration, which is the orange line here. We can see that there was an increased all-cause mortality and an increased risk for several risk factors with underhydration, including obesity, insulin resistance, and hypertension. So overall, our big picture question is, we know that there are race disparities in cardiometabolic health. Could differences in hydration be playing at least a small role here? So that's our big picture question. So that takes us to our first study with the cohort that I mentioned from Auburn University. We were interested in determining whether there were racial differences in hydration using a 24-hour sample as opposed to a spot sample. We only knew of one study that had performed a similar analysis in the past. We're also interested in whether the differences in hydration impact arginine vasopressin or plasma copeptin, and also whether neighborhood deprivation could be a determinant of hydration status. So in terms of linking hydration and health, we know there's a lot of studies that have indicated that elevated arginine vasopressin or copeptin is associated with things like insulin resistance and type 2 diabetes risk. So we know ABP is produced in the hypothalamus and released by the posterior pituitary. And acting on AV1 receptors, it can lead to vascular constriction. So there could be a mechanistic link there to differences in hypertension. So just for anyone who's not familiar, copeptin is a more stable surrogate measure of ABP that's co-released with ABP. So that's what we measured in this particular study. In terms of previous data linking copeptin to race differences in blood pressure, there's very limited data, but it at least led us to want to examine potential correlations here. So this is data led by Dr. George Bagri back in 1997. So this was in 39 patients with hypertension, including Black and white adults. And a subset of them provided blood samples. But what we can see here is if you antagonize the V1 receptor acutely with a pharmacological drug, that you decreased mean blood pressure in Black participants, but not in white participants. And that was largely explained by race differences in ABP. So you can see white participants in gray here, on average, had much lower plasma ABP concentration compared to Black participants. And there was a strong correlation between circulating ABP in the Black participants and the change in their mean blood pressure with the pharmacological blockade. So getting to our study, hydration biomarkers, directionally, there was higher urine-specific gravity in Black adults, not quite significant. There was a significant difference in urine osmolality, and Black adults also had lower urine flow rate from the 24-hour samples. Regarding plasma copeptin, Black adults also demonstrated higher plasma copeptin concentration compared to the white adults. And then we looked at determinants of the plasma copeptin. So as you would expect, there was a correlation between urine-specific gravity and plasma copeptin, and also urine osmolality and plasma copeptin. So we next conducted mediation analysis, given the last couple of findings showing that correlation between USG and osmolality with copeptin. And so in this particular study, I think because of the smaller sample size of 54 adults, the only biomarker that we found was a significant mediator was urine osmolality. So this basically is telling us that we know there's an association between race and plasma copeptin, and that seems to be strongly influenced by urine osmolality. All right, so next we wanted to look at area deprivation and hydration biomarkers. All right, so this area deprivation score that I'm reporting here is the cumulative. So that's the average area deprivation index from each year of life from age 0 to 18. And so we can see a significant correlation with urine-specific gravity. There was not a correlation with urine osmolality. However, there was a correlation with total water intake. So we were able to derive total water intake using NDSR software from 3-day food and fluid records. All right, so a couple other findings that I didn't illustrate, but I wanted to point out, neighborhood did not mediate racial differences in hydration biomarkers. And in contrast to what we hypothesized, plasma copeptin was not correlated with blood pressure, but it was correlated with BMI. All right, so next we wanted to put our hydration data in context. As I mentioned, there was one prior study in college students that used multiple 24-hour urine collections. This was published by Dr. Will Adams and colleagues at the University of North Carolina at Greensboro. And so I plotted their data next to ours. So you'll see UNCG here on the left and NVPL or MyLab's data here on the right. So UNCG is a very different campus from Auburn University. It's a minority serving institute, a lot of first generation students. And I have a collaborator there, Dr. Jess McNeil. So I'm very familiar with their cohort and kind of the health behaviors of their students compared to the students we've had in our lab. So they did exhibit a little bit lower urine volume, not necessarily surprised at that, but similar trend in terms of the race difference. Urine specific gravity values were very similar, and urine osmolality, same overall trend as our data. So that was reassuring. And then next, as I mentioned early on in the talk, our lab is really focused on dietary sodium. So after conducting this study, we became aware of the subset of participants in NHANES that had 24-hour urine collections, and kind of buried in one of the papers, they have urine volume. So we're able to pull that from NHANES in this sub-study, and similar trends there as well. So that was reassuring. And then I've superimposed our data on top of the NHANES data. So You can see we had a little bit higher urine volumes, but overall similar data. That takes us to study number 2 from this cohort, and here we're combining data from Auburn University and our collaborators at Georgia Southern University. So in the first study, we established race differences in water intake and hydration biomarkers, or at least urine osmolality. We did find that neighborhood deprivation seemed to be associated with water intake and urine-specific gravity. And our question here was, do race differences in fluid intake contribute to the race differences in plasma copeptin? So for this, we wanted to combine data that we had available from 2 different cohorts. All right, so we characterized different categories of fluid intake and once again measured plasma copeptin. My PhD student at Auburn, Nina Studi, led this study from the Auburn side, and then Greg Rosicki, a collaborator of mine who was at Georgia Southern at the time, led the cohort at that university. All right, so one of the reasons we were interested in this question was data from Brooks et al. That was published about 10 years ago now, and it looked at the different proportion of fluids that people take in based on race and ethnic group. So for this question, they're looking at the percent of the sample that consumed the given beverage within the preceding 24 hours. Right, so for tap water, we can see about 60% of white adults consumed tap water, where it was less than 40 for Black adults and Latino adults, about 50% for other. And then the data is kind of flipped bottled water. So more reliance on bottled water for the racial and ethnic minority groups, and similar trend here for sugar-sweetened beverages. And that finding was also present for those with high versus low socioeconomic positions as well. So this is an overview of who participated in the study from the 2 sites. In total, we had cross-sectional data from 86 young adults. We were able to replicate our previous findings in the combined cohort. So you can see Black adults exhibited higher plasma copeptin concentration. And then for the drink categories, again, this was derived from our 3-day food and fluid diaries. So we looked at total water, sugar-sweetened beverages, dairy and dairy alternatives, so things like almond milk, 100% fruit juice, diet soda or other non-caloric beverages, plain tea and coffee with sugar or sweetener or cream, alcoholic beverages, and then total fluid intake. So for our findings, we found that white participants consumed more water. There was no difference in sugar-sweetened beverages or dairy alternatives. Black participants consumed more 100% fruit juice. For non-caloric drinks, there was no difference. White participants consumed more plain coffee and tea, and also more coffee and tea that was caloric, so containing sugar or creamer or things of that nature, and more alcohol, which led to white adults consuming more total fluid. So we did find a correlation in this sample between water intake and plasma copeptin. And then we conducted multiple regressions here. So in model 1, we include study site, biological sex, BMI, race, and age. And you can see there was a significant independent association between race and copeptin in model 1. And then in model 2, you can see the standardized beta coefficient goes from 0.51 to 0.37. And race is no longer independently associated with copeptin here. So that results in a 27% attenuation, indicating that that difference in total fluid intake contributes to the race difference in plasma copeptin. A couple other points here. We did combine some social determinant of health measures across the 2 studies for same domain, so things like family income and experience discrimination, but we had to use 2 different questionnaires across the site. So that was a limitation. Maybe that's why there wasn't an influence of either of those measures in this particular cohort. All right, so study number 3 from this cohort is kidney stress. So we're calling it kidney stress. My colleague, Zach Schlater, was here last month and talked about kidney injury markers and acute kidney injury. So in the context of this young adult sample, we're basically looking at kidney injury markers, but we acknowledge that these participants don't have acute kidney injury, but we're viewing it more as subclinical kidney stress, where an increase in some of these biomarkers might predispose to future elevations that could be closer to, contribution to kidney disease. So we know a number of these biomarkers have been developed as being more sensitive than serum creatinine, and they're associated with incident kidney disease, progression, severity, and outcomes in severely ill patients. So I'm introducing a few that our lab has used and that I'll present on in the next few slides. So KIM1 is a proximal tubular injury marker. Nephrin is supposed to represent glomerular injury. NGAL is a general tubular injury marker. TIMP2 and IGF-BP7 are cell cycle arrest markers, and their product has been used as an FDA-approved biomarker of kidney injury. So again, we're interested in this kind of subclinical measure of kidney stress. We know that these biomarkers are associated with the transition from AKI to chronic kidney disease, which affects 15% of the US population. In general, just general chronic kidney disease, there's not a very large race disparity. We can see a small difference here, but the progression of chronic kidney disease is accelerated in Black adults. And I'll present some data in the next slide that there are disparities in end-stage renal disease. And then again, wants to just kind of point that out one more time. We know that these are young adults. We're not saying that they're at increased risk for CKD right now. As you can see, there's a large age-related increase in CKD incidence. But over time, we do know that with aging, Black adults do have a higher proportion of end-stage renal disease for both male and female individuals. So previous applied physiology studies that have manipulated hydration experimentally have demonstrated some association between hypohydration and these kidney injury markers. So one example would be data that my colleague Joe Watso collected for his dissertation study. So in this particular study, we used a euhydration condition. So these would be values for a 70 kilogram individual. So you can see we gave them 1.6 liters of water to drink every day, or that's what they were prescribed to drink. And then for the hypohydration arm of this study, there was a progressive decrease in fluid consumption over 3 days, followed by a 16-hour hydration restriction period leading into data collection. And there was 24-hour urine samples leading into the data collections as well. So Joe was interested in blood pressure monitoring. We use microneurography to assess sympathetic outflow and autonomic control of blood pressure regulation. So he has a series of papers that he's published, but for the sake of this talk, we did a secondary analysis led by Christine Damier, Joe's current PhD student. And so in this study, we looked at young female participants and older female participants. And so you can see that in contrast to what we expected, there was not an increase in NGAL with hypohydration, but there was an elevation in cystatin C, which can also be used sometimes as a kidney injury marker, as well as nephrin and the product of IGF-BP7 and TIMP2. Chris Chapman, who was a postdoc at the University of Oregon at the time, also completed a study where they did a 24-hour fluid deprivation protocol, and they took spot samples before and after the protocol. So just to orient you here, it's all participants, male and female participants to the right of that. And this is their data indexed to urine osmolality. So in their study, similar to what Christine found, there was a decrease in NGAL. However, there was an increase in urinary KIM1, urinary TIMP2, IGF-BP7, and the product of IGF-BP7 and TIMP2. So that takes us to our recent data that we've been collecting. This is data in review at AJP Reg. It's led by my lab tech and incoming PhD student, Jake Muma. So when we looked at absolute urine concentrations, we can see that there was an increase, or not an increase, but elevated NGAL in Black compared to white participants, TIMP2, IGF-BP7, and the product of IGF-BP7 and TIMP2. So this wasn't necessarily a surprise to us because, as we've shown in the preceding slides, Black adults had more concentrated urine. So we then indexed our urinary kidney injury markers to 24-hour flow rate, and that seemed to attenuate a lot of the race differences in these kidney injury biomarkers. So NGAL was no longer different. TIMP2, IGF-BP7, KIM1. We also measured thioredoxin-1 in this study as a measure of oxidative stress. And the one biomarker that remained different was TIMP2 times IGF-BP7. So we're interested in whether differences in hydration contributed to that difference. So we looked at normalized water intake. So here, this is water intake normalized to body surface area, and found a nonlinear association between water intake and TIMP2 times IGF-BP7. This is just zoomed in for individuals who are below that threshold of 0.3, which is the FDA threshold that's used. And next, we broke up the participants based on those who were below 0.3 and above that 0.3 threshold. And you can see those above the 0.3 threshold reported lower normalized water intake. And this is not significantly different, but I think it could be meaningful. About half of the Black participants were above the threshold, whereas only about 1 in 3 white participants were above that threshold. Lastly, we conducted a mediation analysis. I won't show it here, but we also did some regressions to look at that association between race and TIMP2 times IGF-BP7. And we did find that there was almost full mediation by water intake contributing to that difference in IGF-BP7 times TIMP2. All right, so limitations. There are a number of limitations with the data I presented today. So this was a convenient sample of college students, only white and Black college students. As I pointed out with the NHANES data, there's also some disparities that seem to affect Latino adults too, with their trust for municipal water systems and overall hydration status. In this cohort, for the sake of time, I didn't present all that data, but there are a number of differences in some of the health behaviors, things like dietary potassium intake, fiber intake, multidimensional sleep health, and moderate to vigorous physical activity. We know that area deprivation index is a great measure for being a composite score of neighborhood exposure, but within any given neighborhood, we know there's going to be a lot of heterogeneity in terms of family income, family net wealth, the social circle that you're exposed to, and things of that nature. And this was a retrospective study, so we had people give us their address from age 0 to 18 and then kind of looked back to see how that impacted our data cross-sectionally. Another thing that we didn't capture in this particular cohort was information on participants' hydration knowledge or beliefs. And that's something that we're kind of trying to address now. So the idea here is that we know that there are differences in some of the hydration biomarkers and total water intake. What we don't know is whether differences in knowledge about how much you should drink or how you perceive water to be important or not may influence that. So we developed a questionnaire of 13 different items that some of them were developed alongside Sophia Sanchez, PhD in RD in our lab, and also previous questions that were used in some of the CDC reported data. So from the 13 questions, we came up with 4 different constructs: perceptions of water safety and quality, health beliefs and perceived knowledge, behavioral regulation and self-monitoring, and barriers to healthy hydration. And so this is data that was presented by an undergraduate student in our lab, Jenna Michael, at our recent School of Public Health Research Day. So since this particular presentation, we've brought in more individuals, and we're currently measuring osmolality in our 24-hour samples. But even from this really preliminary analysis, we can see that there was a correlation between the subscore barriers to healthy hydration and urine flow rate, and the total score from the questionnaire and urine flow rate. So that is something that we view as promising and that we would like to look at in the future to see if there are race differences in some of these perceptions and whether that may contribute to differences in total fluid intake and some of the hydration biomarkers. All right, so lastly, additional implications. Again, this was a cohort of emerging adults. We'd like to extend this work to middle-aged and older adults, where there could be more robust differences in like hypertension prevalence and some of the other health metrics that we're interested in. And one of the things that we're interested in here is you can see the tree canopy cover. This is images from NASA. So the darker green would indicate more tree canopy cover. So typically in neighborhoods with higher socioeconomic status, there's going to be more tree canopy, more green space. And so we can see across a given city. So this is Los Angeles as an example. There's large variability in temperature exposure. And you can see there's this inverse relation here with the tree canopy cover. And so that kind of led us to wonder, could this be a chicken and egg type issue where maybe in some cases, the temperatures that you're exposed to could contribute to differences in hydration biomarkers, but also if we have certain segments of our population that are already less hydrated, and then also more likely to live in these areas with higher temperature exposures, could that exacerbate problems with things like heat illness or heat stress? And then efficacy of hydration interventions to potentially address some of these issues. So Nina, who I mentioned earlier in the presentation for her dissertation, is doing a really small open label trial with water and potassium supplementation. So that's something that we're interested in examining at a larger scale in the future. All right, so I want to thank everyone who's tuning in now, everyone who's here in person, my former lab members, current lab members, and colleagues. I'm sure I forgot a few people, so I apologize for that. But I want to thank everyone and funding agencies, my dog, Sophie, my dog Chaos, and my wife Sophia for keeping me sane. And I'll take any questions, and I've got my contact information here for anyone who wants to connect or has questions after the fact.
All right, awesome. Thank you. Great presentation. Very thought-provoking and of like millions of questions and ideas. This is obviously a very, very interesting topic. So for the online participants, please add your questions in the Q&A and we will address them given the time availability. Are there any questions here from the room? Mason?
Thanks for the shout out one and appreciate you being here. Real quick, I'm interested, saying speak up or, okay. Real quick, I'm interested in the neighborhood deprivation index do you have any idea of the Black individuals that may be living in a higher socioeconomic background if those trends still remain, or do you know if that's the case or not? I know there was you presented a association graph. I couldn't depict if that was how much I agree with Black individuals or not.
Sure. No, that's a really good question. As I mentioned, so there's 54 people in the sample who had available data, basically. So for the correlation, we combined everyone in the cohort. During peer review, we have had a reviewer ask that same question. Was there a difference in the white adults and Black adults? So to address that reviewer's concern, we did, explore those associations, and they do seem to be more so driven by Black adults than white adults. So that would suggest that maybe it takes being at a certain threshold to kind of start driving those correlations between some of the different biomarkers. That also indicates to me, though, that because the strong correlate, or because there was a moderate correlation, more so specific to Black adults, that those who were more well hydrated didn't necessarily have like a higher copeptin. So this to me seems more of an issue that could be addressed by increasing fluid intake, decreasing urine osmolality, urine specific gravity, and that difference in copeptin probably wouldn't necessarily exist. In terms of the ADI measures, that was a little trickier. I can't remember off the top of my head for all of them, because the way we look at ADI, for sake of time, I just presented the cumulative, but we break it down by early childhood, middle childhood, adolescence, and then total. And I know there were select correlations. Some of the times there weren't a correlation, again, just because it was a small sample size, only, like 20 and 34, or 24 and 30.
We have a question from the Q&A section. Do we know if there have been any interventions to improve hydration at the community level?
I've seen interventions to improve hydration, but there wasn't necessarily a focus on race per se. I have seen, if I recall correctly, there was one intervention that had a high proportion of participants who were Black adults and racial and ethnic minorities, but they didn't necessarily look at like, was there a difference in the efficacy of the intervention between Black adults and white adults? But I'm aware of a few different interventions that have reduced copeptin, or there's one study in Japan where they found there was a reduction in systolic blood pressure with the intervention over 12 weeks. So that's kind of some of the background information that informed our question as to whether this could be something that might at least play a small role in helping with racial disparities in health.
Excellent. I have a question for you. You mentioned one of the first studies that you presented was the hypertonic saline infusion, which I've used it actually over the years as well. There is always when you do the hypertonic saline infusion, you impact multiple things. So one of the things that I'm interested in, I've used hypertonic saline, is to induce the osmotic stimulation of vasopressin to study the impact of vasopressin. And being more interested in vasopressin, we almost ignore or say nothing happens with it's not the sodium effect per se, it's the vasopressin effect. And then also you play with cell volume because you create a hypertonic environment. So you shrink cells, and that could be on its own. Could you speculate a little bit? I mean, it seems that there is distinct impact of all those things probably. Can you speculate a little bit on what's the true role of sodium? What is the role of vasopressin, and whether there is a role of cell volume in this findings?
No, that's a really good question, and multiple valid points. For that particular study, we did measure the renin-angiotensin aldosterone system hormones, and so this is something that's well documented in the literature. But our Black participants tended to exhibit reduced concentrations of like serum aldosterone, plasma renin activity, and so over the course of the infusion, there was a reduction in some of the components of RAS, but the Black participants were not able to suppress RAS as much because it was already, suppressed to begin with. So that's one observation that we did have from the study. We did measure norepinephrine. Unfortunately, we didn't measure ABP or copeptin in that study. We have done some lactated ringers and like other controls in some of the other studies in Bill's lab, but in that particular study it was just the hypertonic saline infusion. So, I can kind of speculate on RAS and the sodium angle, but not so much on ABP.
One more question from online. Thanks for the presentation. In thinking about the association of hydration and the health risk, a primary solution to address this is changing fluid intake behavior. However, strategies that have been used previously have failed to reach scalable and sustainable response. So how do we as scientists in the space that we work, we can change behavior among those not meeting adequate fluid intake recommendation? So I guess it's a big question. Like,
yeah,
it sounds great. How do we change some of this behavior?
Yeah, in our school, we do have a lot of individuals who do implementation science and co-design, and I think this could be an area where if there's been a number of attempts to try to address a certain behavior and the community's not responding per se, it might be a good idea to do some co-design and try to figure out what are some of the barriers that people are facing to adopt the certain positive health behavior. That's what I can think of at the individual level. I mean, structurally, we know that across the country, there's a lot of aging infrastructure, like not just water pipes. So, that would be great if we could address that. I think that would be the most proximal issue, that people trusted the water that's coming out of their tap. I think they would probably be more likely to drink it and then use it to make, other solutions or teas or coffees. But I guess, for us as physiologists, maybe working with, some of our collaborators in like implementation science to try to figure out what we can do to help individuals uptake those positive health behaviors.
Yeah, thank you. I tend to believe that improving or reaching adequate water intake, it might be one of the relatively easier messagings, I guess, that we can do compared to do more exercise, eat less sweets, eat less fat, all the other stuff that we try to, inform people to live a healthier, better life.
I tend to agree. A lot of our work with sodium, I think you're asking someone to take something away, like don't eat this food, don't eat that food. And I think that's what people struggle with. But with water, it's more like, hey, just drink an extra water bottle per day. I think the only real barrier I've experienced personally, I'm talking to people is just like more frequent urination. They just don't want to. But yeah, I feel like it should be easier than like diet modification, for example.
Have time for one or two more questions.
That's another question. So obviously your work focuses on cardiovascular disease and hypertension. However, cardiometabolic disease and hypertension or cardiovascular disease tend to be hand in hand. Do you have any additional data that might implicate some sort of cardiometabolic measures, maybe body composition, for example, or some glucose measures, or maybe even obesity measures in your participants, and how, if there's any relationship between perhaps obesity, anything in the cardiometabolic realm, fluid intake, and race?
Yeah, so we did see the correlation with BMI and copeptin in that particular cohort. I mean, and you know this well, at Auburn at the time, there was limited access to the DEXA room because of the COVID precautions. So we didn't have DEXA in that particular cohort. So I wish we did have, you know, some better measures of body composition. We do have waist to hip ratio. I don't recall if we looked at any associations with copeptin per se. Some of the interventions that have been used in the past in like middle-aged and older adult cohorts, they did find some reductions in copeptin and resting blood glucose. With fluid intake interventions, like extra, like 1.1 liters per day or something like that. In our young adult sample, we don't see any sort of potential race difference in circulating glucose or HOMA-IR, and most of the values are like pretty normal or low to begin with. So I don't necessarily think that they would improve with hydration intervention, but maybe in middle-aged and older adults, there could be some room for that. So
I have one more question. So I've been following the publications from Natalia Dimitrieva from NHLBI, from NIH, that she has been dichotomize data from population data based on plasma sodium. And she's using that as an index of hydration, which it is reasonable in my brain. However, high or low sodium. So what I'm trying to ask is how confident you feel using this type of classification based on plasma sodium, knowing that high or low sodium diet could impact sodium concentration, what we're measuring in the blood.
yeah, so Graham McGregor and his group have a lot of data where increasing sodium intake in the diet can lead to small changes in plasma sodium. I haven't seen a lot of large scale data linking habitual sodium intake to plasma sodium. So these, what I'm describing, the more like intervention studies that are at least coming to top of mind right now, where over the short term, if you increase sodium, you increase plasma sodium. But I know like some of the collaborators I've had in nephrology are also kind of that same opinion, where the plasma sodium or serum sodium is more reflective of hydration status as opposed to dietary sodium.
I see the beauty of this biomarker is that everybody's measuring it.
Yeah.
Versus, I don't know, all the other stuff that we routinely measure in our laboratories, like osmolality and all of that stuff.
Mm-hmm.
Nobody does it in cohort studies. We don't have plasma osmolality measured from enhanced, it's calculated. And so the practicality, I think, makes it very attractive for me personally, and as a message when people do routine blood exams. See, I think we have one more question. Do we know if the racial disparities in hydration persist into older age, or is it something primarily for younger adults?
So the NHANES sample that I presented on, that was a fairly wide age range, but at least like the central tendency was quite a bit older than what we've looked at. So I would say it does persist into middle age and older adulthood.
Awesome. So as a closing, I'd like to ask the question, what do you think is, like, if we had and limited funding, something, what is the question that you really want to ask or answer more appropriately, or what would you do as a future in this area of research that you've been doing the last few years?
Yeah, I think one would be kind of building upon the work we're doing right now at IU. It's just my lab. We're a fairly small lab, so we're trying to bring in a little over 100 adults, and there is actually a fair amount of kind of ethnic diversity in this sample that we're collecting, but there's not a lot of overall racial diversity, particularly with African Americans, just given the nature of our campus population in Bloomington, Indiana. But to expand that out and look at some of these perceptions and fluid knowledge and how that is associated with the hydration biomarkers in 24-hour samples or even multiple 24-hour samples, I think would be really interesting, like in a very large sample, to see, how is that explaining some of these differences. And then separately, I'd really like to do an intervention study because it's well documented in the United States that there's also health disparities in potassium intake. So we know that it's not just sodium, but other things that you eat in the diet alongside of that sodium. So this is very well documented that there's race differences in potassium intake, not necessarily sodium intake. And so one thing that I've been interested in for a while would be like an intervention where we look at increased water and potassium, or just potassium alone, and see if there's potentially an additive effect or maybe a more beneficial effect of like the potassium or the water alone, versus one of the others in isolation. So if I could do like a large intervention like that was well powered, a large team, large sample size, and everything like that, that would be really interesting to me.
Awesome. Thank you so much. Really appreciate for the really interesting talk. Thank you.
Cool. Thanks, everyone.